where we will be picking up where we left off. So I'm going to start as usual with a sort of a recap of what we did last week. Maybe we can go directly to some of the plots we did. So some of the so the whole project, starting from the beginning, is it starts with this observation that when we have genomes that come from Oxford Nanopore in particular, they have this error profile where what we see is the more indels compared to the standard error profile that we had from Illumina genomes, especially MAGs, where what we would see is that the genomes would be broken up and fragmented. And so when we have these indels, the downstream effect is that we have mispredicted genes. We have genes that are cut a bit too short or outright missing. And so we thought that, okay, maybe we can use these broken genes, and we've never really gotten a good term for them, but these apparent pseudogenes. So a pseudogene is something real, biologically real, that looks like a gene, but it's not a gene. And here what we're interested in is things that kind of look like a gene, but they're not real because they're sequencing artifacts. So if we thought if we had too many of those, that's probably a sign that something's bad. And so we decided to, okay, let's see what we can learn from this. So we started with simulation, okay? And here, let me just change. Start with simulation, and what we did was we started with these genomes, and for example, we took E. coli K12, and E. coli K12 should be some sort of weird thing with the streaming. So E. coli K12 should be very high quality, and so we measure how short are its genes after grouping them by KO. We had a few weeks where we went back and forth on what should we use as a reference. So we settled on the GMGC, and so we can get for this. So there's some technical thing here, but I think it should be fine. I slightly changed the streaming settings. So we have a genome. We find a gene. We classify it as KO123, and if that gene has 300 amino acids, we use the GMGC V1, actually only the complete genes, and to go from 300 amino acids to a z-score. So if the average size of that KO is 300, then that's exactly 0. If the average size is smaller, then that will be a positive z-score. And if the average size is larger, that is a negative z-score. And so what we saw was in different ways is summarized in this picture. So this is the distribution, so the cumulative distribution of z-scores. And so blue is the wild type, 100 is 100 mutations, and 1,000 mutations, and we see that there's this gap, and we were trying to figure out what can we do. Can we get some insight into this gap, or this was the initial, some validation of the initial intuition. And so we spent a few weeks then trying to see if it's possible to clean up the data. Maybe some KOs are not very good. They have, they don't have stable sizes. Maybe some of the alignments are not good, but it actually seemed that in some instances there are real. It's hard to. We couldn't really, or I couldn't really find a rule there, and so last week we said, okay, maybe we just take everything and we look for this excess. And so and since I was a little bit lazy to do the math on how to properly normalize and do area under the curve, I just took the sum of the Z scores below, and I said -4. And it was just a very hacky way of doing things, but potentially, at some point we can try to do it properly. And so the result was that, if we do this, this was the final result. Let me here. So each curve shows an organism. Each on the x-axis we have the number of mutations. Again, this is all simulated, and here the dashed line shows the value at 0.0. So because we can use, I forgot this word on stream, we can use the high quality genomes to calibrate the thresholds. So for example here -500. So if in this version of this score a genome scores at -500, that seems to be worse than any of these, admittedly 5 genomes that we had looked at. Right, so this is where we left off. We have a draft of something that looks like a score, in the sense that, given a genome, I can compute this, just do a little bit of math, and we can compute this. And it seems that at least when the number of mutations is high, it achieves some separation, right? But okay, there were 2 things that we really wanted to look at today. So one is, right, we only have 5 genomes, so not great. And the other one is we left the code in a little bit of a messy situation last week, so we want to go back, clean that up as well. All right, so let's start there. Let's start by just refreshing where we were in terms of the code. Okay, normally I work here, source, and everything is also in this work in progress directory, work in progress branch. And ideally we would like to clean this up and move it to the main branch. So how do we do this? Let's switch back to main. Did I leave some? Oh, okay. I actually. Oh, I forgot to switch main. Let's move back to the main. I'm gonna just quickly. Link. Okay, so the this is correct. Yes. Okay. So I just got that. There's probably a way to do it with Git, but this works. So I just got so now. This is where we left off last week. Right? So and the other thing we talked about last week is that the code is now also a little bit slow. So ideally we would want to pre-compute some of these things. All of this stuff is fine. Probably at some point should be moved to a different. Okay. Okay, so we've just been adding code and code to this. So at some point we should really for example, this sort of logic, this let's move this into the okay, and I think it has to be like this because this column is going to depend on that one, so I think we need to have it as a two-step. I think this is correct. Okay. Okay, so that's slightly better. Okay, all right, so we'll do this. So this is slow. Can we make this a... I'm not sure that... Yeah, I think this is fine. Doing the... So, okay, so we move... So we loaded the diamond. We looked at all the mutations. For each mutation, we build this data. What does data contain? And then we build the z-scores table. Okay, this is a bit slow. Yeah, okay, this seems a bit atrocious. I see. Okay, so this can be okay, so we have AA sizes like AO. What is this? So we can do something like this. Wait a minute. Oh no, it's not keep index. It's wait a second. I got this. Okay, something like... Okay, we can do something like this. Okay. Okay, so we can pre-compute this. So we can pre-compute the sizes of KOs. KO sizes. So this can be pre-computed and move outside of the loop here. And now this z-scores that we did. Okay, let's see. Yeah, this is. Okay, so we did this. Okay, so we can do a bit better here. Have the data, and we can join with the KO sizes. Okay, this, give me a second. This will. Yep, some. Yep, this works. Yeah, something like this, except it's more like something like this. Yeah, I think. This is the same. Okay, we called it Z. Okay, something like this. I think this is the same. This takes a. Okay, this is. So this is also a pretty typical way of doing it. So we've a pretty typical way of doing it, which is we first wrote code that was kind of ugly and or slow, sort of doing this by hand. You know, this sort of iterating these polar tables by hand is terrible. But now we can quickly, we can do it. We can use that to we can use that to check that this new code, which runs in a few seconds actually, computes the same thing. Yep, okay. Okay. Well, so this is much faster and also much nicer, generally speaking. Just be a little bit more consistent. Oof. Okay. Okay, we call it number mutations. This avoids. Okay, this is fast. So this also we probably could have done a could have done kind of group by. No. Up. Sorry, this is my bad filter. Select number Z. What's it? Yeah, we can. Okay, we'll get the same like this. Since it's fast enough, let's forget about all of that. Okay, this works well. Okay, I think this is good. Let me just something. Let me just adjusting the camera here, but yeah. This is good. Oh, sorry. It's now still going to take a few seconds to do all the figures, but it's probably going to be much faster. So this was several minutes before. Okay, there they are. Sorry, my bad. Several minutes before. Yeah, okay. Okay, so this is good enough. I'm gonna call this good enough. I think, yeah. I think that was. I'm gonna call this good enough. I think we've managed to make it significantly faster, clean up a little bit, refactor the code. Factor the code, slightly easier to understand, made it, got rid of the slowest bit. Just run it again. Okay, this bit still takes a little bit of time. Parse all the, but it's okay. I think we're let's in the interest of time. Okay, so this is now finishing last week's work. Good. For extra. Likely going to. So this is likely going to be a. There's likely not going to be the final version. This idea, but it is. It does seem separate. Okay, so this wraps up last week's session with a bit of a cleanup. And now what I promised today is that we would look at a few more genomes. And actually I wanted to do two things. I wanted to, so far we've, okay, I did pre-prepare a little bit. So far we've been looking at, let me go here. So we've downloaded data. So we've downloaded these from ProGenomes, and kind of chose a few. I don't know. So we can just increase. These and potentially, I guess eventually do them all. That would probably be a bad idea. At least, I don't know, maybe not. But the other thought I had was looking at genomes from Spire and just looking at some, taking some, maybe not all of the representative genomes, but maybe looking at some genomes from Spire. So Spire is a collection of metagenome assembled genomes. So there's quite a few of them, over 1 million. And so we could use this. Oh, look, for some reason we could use this. So for example, we could take, actually, okay, I guess this is not how it works. Let's take, okay, I guess. Oh, I actually thought my data was there. It's associated. What's the... Okay, actually, I thought that... Okay, I don't know why I was not finding it. Maybe I was spelling it dogs instead of dog. No. Okay, so I'm gonna get them. Let's get these mags. And so the advantage here is that, so these are Illumina mags. So these are gonna have slightly different error models. And so okay, so let's just download the data. And actually, so today is 11. Okay, so cleaned up from last week. Okay, this is here. So I think this is only partially there, but okay. If I recall correctly, I think if you download this like this, you're actually getting all the mags and not just the filtered versions. But we will actually know that in a few seconds, or not in a few seconds, but we will know that at some point in the not so distant future. All right, so we've gotten those mags, and then I don't know. Let's just again. Just pick some. Let's pick some species that we like. This is how you're supposed to do it. Is that correct? I'm gonna use okay, this is it's not how you it's not misspelling. Okay, let's get something. Sorry, let me. Like sorry, let's. So this is. Oh no, sorry. I'm sorry. Is it candidate biology? Did I misspell? Okay, sorry. Some of these are. I think it's the website that's not working very well. Biology vector. I don't know which one would be the. I don't know. Okay, at some point we need to do it. This is not really great use of time, but what's 1 or 0? Okay, this is not. Okay, 1 is E. coli. Is that what we had before? Let's see. What's 2? It's pretty slow. I don't like this. Okay, this is what I was looking. What is the representative? How do I see the representative? Okay, this is. This is the one? Yes, this is the one. Okay, what's 3? That's good. Is this what we did before? Campylobacter. That's probably Staph aureus. Alright, let's skip 4, guessing it's Staph aureus, and go to 5. Streptococcus pneumonia. Alright, that's I don't know why Copilot keeps making up. Okay. Okay, we're still thinking whether we should be a bit more aggressive with this and just download everything. All right, tell you what, let's do this. Yeah, let's do this. Let's download everything for future use. Today we won't look at that potentially. But we will have the data downloaded for next time. Okay. Expanded. This is, yeah, and then we downloaded this. As I said before, part of the goal of me doing this in general is that we are dog fooding our own projects and our own data, and so people should be able to do what I'm doing. And so if I find issues, that would mean that there things should be improved, and I'm finding it now. So the dog fooding, we using our own tools is part of what we, part of our practices to try to get good quality tools and good quality science. And so, yeah, so I am almost always trying to, see if, people should be able to use Progenomes 3 for this, so I should be able to use Progenome 3 for this. People should be able to use the mags from Spire and that we have from our paper, and they should be good. So I'm going to be trying to use that. And if I run into any problems, maybe we can fix it for everybody. Expand dataset of genomes. Okay. So if we now go back to the source data, okay. Okay, so we have 3 new ones, which I would now have to look up what they are. So this is correct. Not a bad guess, but it's Fusobacterium. Yep. Okay. So this is probably not going to finish, so we'll start wrapping up in a few. But this is probably not going to finish, but let's just see. Okay, great. Oh. Okay, yep. Okay, so this is nice. Okay, this is nice. So this is, let's see. So what's happening here is that for at least one of these genomes, it's not okay, so the prevotella-copied genome is not a single. The prevotella-copied genome is not a single contig. And so, which is obviously okay. The only issue is that the code does not handle it properly. So we could always do this. Probably a bit extreme, but could always do this. Huh. So obviously this would also be a huge issue for processing the mags, because the mags are not something that we can that are going to be heavily fragmented. Okay, so this is, but let's do it this way. No, sorry, I didn't mean to actually. Okay, so add. Okay, this is, okay, I'm gonna just add this to the, but will not forfeit one. Okay, so this is, yeah, so we're still downloading the data for everything, but this should just run, and we should be able to see the plots for a couple of these extra. We have a few more minutes. Oops. Okay, or maybe not. Okay, maybe not. Okay, something here. Okay, so And let's just debug this real quick. Oh, I see. This is this was okay. This was bad. Okay. Okay, so I just messed that up. Actually, very bad. Oops. Okay, now. I'm trying to wrap up this session because we're slowly running out of time, and instead I keep doing stuff that is kind of nonsense. I might have to kill this. Okay, right, let's just let me slow down a little bit. Okay, so we got this. Get the sequence. Okay. I need to fix the okay, so the only thing we changed is we fixed reading the headers. Okay, this looks more like what we wanted, right? Since we messed this up a bit, let's do this. Okay, this is what we had before. I'm gonna clean up. So anything that was outside of that specification or is gone, and right, and now we execute, and it should run. Okay, protocol's running. That's so things. Okay, so now it seems that it's great. Okay. Okay. Okay, so this is gonna run. As usual, I will let this run, and then when I do the recap, I will show what the outputs are. In the meanwhile, so it's also we downloaded the mags from the dog study, which we will start looking at next week. We will also probably have to deal with the fact that, so before we had 5 species, and so we had a 5G of this output directory. That's not really going to, that's really not going to scale. So even if we see, 500 M for this, probably not going to scale. In fact, we don't even want this. All of this, we're just parsing this to get the headers. So probably what we're gonna do is we're gonna run it, and then just parse to get the headers, get the information we want, and throw away everything else. Also add a bit of compression. So we'll have to do a bit of that type of cleanup as we switch to working with a whole representative data set. Okay, in the meanwhile, all right, now we are going to finally really wrap up. Stream notes. Okay. Okay, next streams. Nope, this is not. Put it. Okay. Okay, so this is it. Summary of workday, and okay, so I'm just pushing it now. So right, so there it is, as usual. So everything is on the GitHub. So in this case, I've pushed to the main repository. Everything's there. So I will post the results of running the code we wrote a little bit later in the week during the recap. So I see there's a question in the chat I just noticed. That type of question, please just send me an email. We actually do have some PhD opportunities if you're interested. So just send me an email. It's more of a one-to-one discussion than something to be had in a large group. But yeah, so join there's a mailing list. There's a Discord. Please join us there. And I hope to see you here next week. And I will be